Welcome back, folks. We've been talking about membrane structure, function, and transport across membranes, and so let's keep thinking about that. Now, one of the major functions of the membrane, which arises from its selective permeability we've started talking about, is the ability to maintain a gradient. So I've already talked about gradients a little bit. Gradients can be there can be a couple of different types of gradients. So let's start off with the simplest type of gradient, a concentration gradient. So on the slide here, what we're seeing is a lipid bilayer, and in the lipid bilayer there are some transmembrane proteins, and those transmembrane proteins aren't doing anything right now. And outside the membrane there are a large number of glucose molecules. And so if this was a cell, this would be the cell membrane. There'd be a lot of other proteins here, a lot of other things going on. So this is very simplified. So outside the membrane is a high concentration of glucose, and inside the membrane it's a very low concentration of glucose. So what we have is a concentration gradient across the membrane, and so there's a high concentration outside and a low concentration inside. So if we think about what the glucose is going to do, what its behavior will be, there's going to be the random movement of glucose in all kinds of different directions. And if the glucose could move, it would diffuse in the net direction into the cell, so that we ended up with the same amount of glucose inside the cell and the same amount of glucose outside the cell. And that's because that's for reasons of thermodynamics. It's more thermodynamically stable to have the same concentration on each side of the membrane. We're not going to get into that too much, but what would actually happen in this case? Well, the glucose really isn't going to go anywhere in terms of into the cell here, because remember, glucose is too large and it's too polar. It's covered in a hydration shell. So yeah, there's a very low chance that one of these glucose molecules could sneak through the lipid bilayer, but that's going to happen at such a low frequency that we would say that the net diffusion of the glucose into the cell here would be very, very, very slow. And it's driven by differences in concentration. You can actually work out the size of the force that would be generated because of the difference in magnitudes of the concentrations in any given scenario. So that's one type of gradient, a concentration gradient, and this is what membranes are able to do. They're able to keep things outside that need to be outside, and things inside that are meant to be inside. And that's the beauty of their selective permeability. Things that we just want to go and come and go freely can do so, but things that need to stay outside because of their chemical properties and because of the chemical properties and the structure of the biological membrane. So what other kinds of gradients can we get? Here we're looking at ions, and so if you pause the video and count out the number of ions, is what you'll see is that there are more positive ions outside the cell and more negative ions inside the cell. And so what that creates is an electrical difference across the membrane. You can think of it as an electrical gradient if you like, but that's called a voltage basically. And so if we have more positive ions outside the cell, more negative ions inside the cell, and that is actually the situation that occurs inside. The cell is slightly more negative than the outside. Is what happens then is that the charges attract one another, and the positive ions, or the positive charges outside, are going to be wanting to move inside. That's a little bit anthropomorphic. They don't want to do anything. They will do that because of the laws of physics and chemistry. And so this is a type of electrical gradient. You've established a voltage across the cells, and all your cells have a voltage across them. And in some instances, that can be used to do work. For example, generating an action potential in a neuron. But all cells have this charge across their membrane. Now, if we look at one ion, let's look at sodium, the purple balls here. You also see there is more sodium outside than inside. Let's look at chlorine. There's more chlorine inside than outside. And so there are also chemical gradients here. So there are two kinds of things going on here. There's the electrical gradient, which is the net effect of all these charges, but then there's a chemical gradient. And so in some cases, the chemical gradient will be driving a solute in one direction, the ions in this case, and the electrical gradient will be trying to drive them in different directions. So for example, the chlorine here, the concentration gradient will make the chlorine want to diffuse out, but the electrical gradient will be trying to keep it in. And so there's an equation that we're not going to use. You use this in general biology too to look at membrane charge differences, or differences in charge across a membrane. It's called the Nernst equation. We're not going to use it, don't worry about it, but it lets you look at the relative effects of concentration gradients and electrical gradients across membranes. And so when you have both an electrical gradient and a chemical gradient, you have what's called an electrochemical gradient, and it's usually this type of gradient which is more important in biology. So those are two types of gradients that you should know about. Now, when we get gradients across cells, we have this language of describing the nature of the gradient. So if we're talking about a chemical gradient, if we've got two solutions separated by a cell membrane, and don't just think about the outside the cell and the inside the cell, we could be talking about the inside of the ER and the outside of the ER, the inside of the Golgi, the outside of the Golgi, the inside of the mitochondria, the outside of the mitochondria, any two spaces separated by a membrane. We're going to have solutes being separated, and a lot of those solutes can't move. So when you have equal concentrations of solutes on the outside, that's called isotonic. Most cells are bathed in a solution which is effectively isotonic. That means there is the same amount of solute outside as the same as there is inside, and we'll talk about why that's important in a moment. So that is isotonic. Iso means equal, and tonic here is referring to solutes. When you have a concentration gradient, one side of the membrane will be hypertonic. So in this case, the outside of the membrane, I'm going to say that's on the left. The left side has got more solutes, so it's said to be hypertonic. So I would say, well, the left side is hypertonic relative to the right side. Hyper means above or beyond. So hyper means there are more solutes on one side than on the other. Now, this is a relative statement. You can't just say, oh, the solution is hypertonic. It doesn't. That's not how the language works. The left is hypertonic relative to the right. Now, that means that you can also have the opposite case, where you can have one side being hypotonic compared to the other. Hypo meaning under or below. If you've ever seen a hypodermic needle, that goes below or underneath the dermis. Hypodermic, below the dermis. Here we've got one side with a solute concentration lower than another side. So the left is now hypotonic relative to the right. So relatively easy, but you should be able to recognize these things. Who cares? Why is it important? Well, these gradients of solutes determine the direction in which water will be forced across a membrane. So lots of places this is important in biology. In plant biology, it's absolutely critical in water movement into the plant through the roots and then through the plant body. In animal cells, it's important that cells be kept in an isotonic solution so they don't either shrivel up or explode or burst. So what happens to water movement when you have a concentration gradient of solutes? So this is complicated. This is probably the first most complicated principle that we've dealt with. I'm going to make another video for this, which is using a different tool, which I'll post in Canvas. But here's what happens. If you have a hypotonic solution, and on the left, that's hypotonic, the right is hypertonic, water will move from hypotonic to hypertonic. So in this case, if we've got a hypertonic solution outside the cell, and the inside is hypertonic by definition, water will move into the cell. Okay, so if you take a patient and you mean to plug them into a bag of isotonic salts to increase their blood pressure, because maybe they've lost a lot of blood or something, and you've got to get some fluid into them, or you're rehydrating them because they're an athlete and they're dehydrated. If you're getting solution into their body, into their blood, if you put in water and not an isotonic salt solution, so a saline drip, if you put in water, is what happens is that all of the fluid outside of the red blood cells in the blood, the plasma, the solute concentration drops because you're diluting it with water. Now we're going back to using the chemistry we talked about when we introduced the idea of concentrations. And I asked some really simple questions with that stuff, and now we're using it. So if I take something with a given concentration and I start dumping water into it, the solute concentration drops. So if you put pure water into someone's blood, the solute concentration of their blood plasma drops. Blood plasma is the non-cell component of blood. The concentration of solutes drops. And so relatively, we get a hypotonic blood plasma. So what that means is water's then going to rush into the blood cells, and then what happens? Well, if it keeps happening, if you don't stop that, then the blood cells will explode. So what we've got going on here is, if we've got blood cells in your bloodstream in an isotonic solution, which they are normally, if we place them in a hypotonic solution, so you basically increase the water you have in your bloodstream, then the red blood cells will explode and die, and that's generally always going to be a bad thing. What happens if we do things the other way around? If we increase the solute concentration by, let's say, decreasing the amount of water in the blood, then we've made the blood plasma hypertonic, more solutes relative to the inside the cell, and water will leave the cells and they basically shrivel up, and we call that crenation. So that's what happens in animal cells, not just blood cells, but all animal cells. What about in plant cells? Osmosis, like I say, is super important in plant cells. If I didn't say it before, osmosis is simply the diffusion of water across a selectively permeable membrane. So what's happening in plant cells here? If you put a plant cell in a hypotonic solution, then water enters the cell, and the cytoplasm and the vacuole pushes back against the cell wall, and the cell becomes turgid. The cell doesn't burst like animal cell would, because it's surrounded by this rigid cell wall, and that is a healthy state for a plant. Now, if you go the other way and you put the plant cell in a hypertonic solution, that, for example, would be what would happen if you don't water your plants. The concentration of the solutes would go up, and what happens then is water leaves. Water would leave to the relatively concentrated solution around the cells, and what happens then is the plasma membrane pulls away, it shrinks away from the cell wall in a process called plasmolysis, and that's very damaging for plant cells. Once that happens, you can't recover them by rewatering them, and they usually die. So if you see a plant starting to wilt, it's in that very early stages of plasmolysis. If it's early enough, you can recover it. And so osmosis, this movement of water into or out of cells across a selectively permeable membrane, is critical for maintaining water balance in cells. So think about our discussion about biology principles. Homeostasis is a governing, or is a major biological principle. Regulating whether water moves into cells, out of cells, that's an important homeostatic mechanism, and cells have lots of different mechanisms for controlling the movement of water by fundamentally either pumping water out, pumping water in, or in many cases changing the solute concentration of the cytoplasm, and that can draw water in, or it can cause water to leave. Okay, so that's a little bit about osmosis based on gradients of solutes across cells, and we talked about two types of gradients. Let's do some questions real quick. I'll leave these up as I usually do, pause the video, and see how you get on with these few questions. Okay, here we go. Okay, here's the next one. Here comes the third question. Okay, so that's the last question. So this has been probably the most complex set of content so far. You'll have to do a little bit more reading with it. I've put a lot of videos in Canvas to help you with this stuff because it's a little bit more complicated. Now, if you're looking around on YouTube and you're Googling this stuff, which you need to do, if you find a video which you think is better or helps along with the ones that I've posted in Canvas, send me the YouTube URL and I can get those added that other people can see as well. So that's it for now, and we will pick up again shortly.